Te hei maare ora, e ngā mana e tēnei whenua, e ngā waka e ngā tai e whā, te whari e tūnei, tēnā koutou, tēnā koutou, tēnā koutou katoa. Ko Ngāti Pākehā toku iwi, ko Greg Cook toku ingoa. It is my pleasure, on behalf of the Vice-Chancellor and the Professoriate, to welcome you here on this very special occasion, celebrating the elevation of Rajesh Katare to full professor. I would like to welcome the special guests of our speaker, both in person and online, his wife, Jaya, son Ashwin, daughter Anushka, brother attending on Zoom, Ravi Kumar Katara, other family members also attending on Zoom. So delighted to welcome you and thank you for being here. Reaching the rank of full professor is a major milestone in an academic career. Our promotions criteria are no small hurdle. To reach this level, the university looks for leadership, exceptional talent, and original thinking in research, in teaching, the ability to truly inspire students and bring fresh ideas and service that shows a long-term, meaningful and distinguished contribution. The bar is high and the process is thorough, with 3 committees and international referees involved along the way. So becoming a full professor is a major achievement. It marks someone as a leader in the field, in their classroom and across the university community. Rajesh, it's a real privilege to welcome you to the Professoriate, so congratulations. Long ago, an inaugural professorial lecture was almost like a test. The moment where you had to prove you deserved the title. Thankfully, those days are behind us. Tonight is about something very different. We are here to celebrate your achievements and to hear more about your journey. We're really looking forward to learning more about what drives you and the research you've contributed. And because we like to make the most of our academic regalia and ceremony, we have several esteemed colleagues with us today. Tēnā koutou. We will begin with an introduction from Professor Julia Horsfield, and after the lecture, Professor Pete Jones, who will thank you for the lecture, followed by a special thanks from Professor Megan Gibbons. You are then welcome to join us in the Staff's Club for refreshments. I'd now like to welcome Professor Julia Horsfield to introduce our guest of honour.
Ngā mihi nui ki a koe, Professor Cook. Associate Professor Gibbons, tēnā koe. Professor Sika Patonu, tēnā koe. Professor Jones, tēnā koe. And Professor Katare, tēnā koe. Tēnā koutou katoa, nau mai haere mai ki te kaupapa o te wā. Ko Julia Horsfield tōku ingoa. Ko o te tumuaki o te kura Biomedical Sciences. Nō re rā, nau mai haere mai. Well, hello everyone, my name is Julia Horsfield and I'm the Head of Faculty of Biomedical Sciences here at our Otaku Whakaihu Waka, the University of Otago. It's my absolute privilege to welcome you to this significant occasion, the inaugural professorial lecture of Professor Rajesh Katare. Rajesh is an internationally recognised cardiovascular physiologist and an academic leader at the University of Otago. He completed his medical degree at the University of Mysore in India, and he earned his PhD in cardiovascular and biomedical sciences at Kochi Medical School in Japan. And this is where he began his journey into understanding the molecular mechanisms of heart disease. Following this, he served as a lecturer in Japan before moving to the University of Bristol's Heart Institute in the UK as a research fellow, and this further strengthened his expertise in cardiovascular physiology. Fortunately for us at Otago, Professor Katare joined us on the academic staff in 2012, and here at Otago, Professor Katare has developed an academic career that exemplifies excellence in research, teaching, and leadership. He was promoted to Associate Professor in 2017, appointed to the Head of Department of Physiology in 2023, and he achieved the rank of Professor in 2025. So over the past decade, Professor Katare has built a thriving multidisciplinary research program conducting pioneering work that bridges basic scientific discovery and translational medicine. His team focuses on uncovering the molecular drivers of diabetes-induced cardiovascular disease and developing innovative RNA-based and tissue engineering therapies for heart disease. And his group has made landmark discoveries identifying early molecular signatures of diabetic cardiomyopathy and creating novel microRNA-based therapeutics and bioengineering scaffolds to regenerate damaged heart tissue. So he's got some remarkable achievements here at Otago. We all really love our research funding at Otago, and I'm happy to say that Rajesh has brought in over $3.2 million in external research funding. He has more than 130 peer-reviewed research publications in leading journals too, such as Nature Cardiovascular Research, Nature Reviews Cardiology, Circulation, and the Journal of Clinical Investigation. So well done, that's awesome. He has over 9,000 citations and an H-index of 50. Aside from being an excellent researcher, Professor Katare is equally passionate about teaching and academic leadership. He's shaped curricula, chaired the Health Sciences First Year Hubs program, and that's no small feat. He's contributed to the MBCHB Curriculum Committee and taught cardiovascular physiology to medical and surgical trainees from all across Australasia. He has supervised 18 PhD students to completion and continues to mentor emerging scientists. Beyond Otago, his influence extends nationally and internationally. He's an associate investigator with the Healthy Hearts Aotearoa New Zealand Centre of Research Excellence, the Maurice Wilkins Centre of Research Excellence, and the Centre for Bioengineering and Nanomedicine. He has served as National Secretary of the Physiological Society of New Zealand, trustee of the Dunedin-based Medical Sciences Course Trust, and advisor to the Indian Society for Facial Trauma and Reconstructive Surgery. He's also quite heavily involved in editorial contributions, including roles with Cardiovascular Diabetology, Experimental Physiology, and Frontiers journals. His achievements have been recognised quite recently, actually, and most recently in the University of Otago's 3Rs Award. And that was my absolute pleasure to be there that night to see you get that award, Rajesh, and fantastic research in substituting mice in labs with people testing their own saliva. So that was really, really fantastic to see. So he's also received the Department of Physiology Research Achievement Award for publishing his 100th paper. And that was back in 2021. And then a Japan Society for the Promotion of Science Fellowship. So Rajesh is clearly involved in excellent contributions all across academia at the university. And in tonight's lecture, entitled A Roller Coaster Journey Through the Molecules of the Heart, Rajesh will reflect on his early life, his aspirations, and the unexpected turn that sent him to medical school and on the path to research. And Rajesh will also share key discoveries from his research that continue to inspire his quest to combat diabetic heart disease. No mai, haere mai, please welcome to the podium Professor Rajesh Katare.
Sure, Julia. Tēnā koutou, tēnā koutou, tēnā koutou katoa. Ko India te whakapaparanga mai, ko St. Thomas Mount te Maunga, ko Marina te Maona, ko Kuom te Awa, ko Chennai Aho, ke Otepoti au e Nohoana, e Aorangi au te Otaku Whakahi Waka, ko Katare te whānau, ko Rajesh Tokingwa, tēnā koutou, tēnā koutou, tēnā koutou katoa. Kia ora everyone, thank you very much for coming in for today's. I didn't feel any nervous until the morning before everyone asked me, are you ready? I think I'll get through this. And thanks to all the colleagues and family and friends being here. Thanks for accepting the invitation. And thanks to my extended family who are watching this from Zoom. And thanks to the wonderful introduction, Professor Cook, Professor Horsfield, and thanks for coming along, Pete, Professor Megan Gibbons, and Professor Dan. Thank you very much. So I was thinking, how do I need to frame my lecture? And I thought, okay, maybe let's do it in a way more of a general and try to keep it as simple as possible. I'm going to talk a bit about research, but also my own life journey. Hopefully it works. Great. Before I start, I just want to start by thanking Almighty God, because I believe that magics do happen. I didn't believe that, but recently I started believing it. Because if you asked me last Tuesday if I'd be delivering this presentation, I would have said no, because for the past 3 weeks, I've been suffering from my sciatica pain. From this last Wednesday, the pain started decreasing, and this is the first week I've been working continuously since the pain. So there's always something which helps us. There's always a power. And my next thanks to my parents, so without whom I wouldn't be here. So I have heard from my dad and mom that I was the last child. I'll show my family photo. And they're really thinking, do I need me? So it's pretty late. But thanks for them for bringing me to this world. And a big thanks to my sister-in-law. Sorry. She was like my second mother, but she unfortunately left us 3 years back. Thank you, Mala Bhabi. Great. I tried to find my younghood photo, but this is the latest one. This is the youngest one I find. This is like when I was 5 years old. And this is when my sister got married. So my sister and me were like 17 years different. And this is my family photo. This was taken in 1983, I guess. I should be around 10 years old. And that's my elder brother. So he's watching online. Is Ravi Kumar. So he is working as a business developer in Corning International in US. My second brother, here is he. His sister, younger brother, Suresh. So he has his own company of IT. He went back to India at a perfect timing when the IT was blooming. And this is my sister and my uncle. So they are here like this now. And this small boy is Bharat. He's in Brisbane now. So he's an IT guy, well settled. And I also want to show one guy this is me again, at some age. And this boy here, he's one of our pet child. He's Rajesh. His name is also Rajesh. So he's right now in my elder brother's son. And he's in San Francisco and working as associate director for a pharmaceutical company. So this is my family. And thanks to all my family, because my journey wouldn't be fulfilled without my family. So where I am from. So I'm from India and Chennai. Chennai is the southernmost part of India, which, as some of you might know, it's one of the hottest city in India. And the summer temperatures are roughly around 40 to 42 degrees. And you could see how we feel when we came to Dunedin. It's a good city, big city. It's more modern. It also has a lot of culture. So it has a lot of temples. Indians, we love temples. There's a lot of history to be associated with that. And it has the longest beach I would say, second longest beach in the world, marina. And crowd, people. So Chennai has around 14 million people in population, so which is almost three times more than New Zealand. Crowd, people, people, people. This is just a big shopping street where you find so much people just before a festival. And if you're following cricket, that's we, Chennai Super Kings. And Chennai Super Kings is very close to New Zealand, Kiwis, because Stephen Fleming is continuing to be the Chennai Super Kings chief mentor, and along with Dhoni, and they have been doing so well. So on Chennai Super Kings, we love Chennai Super Kings. A lot of our Kiwi Black Caps have played for Super Kings. So that's Chennai. And what do we do? Because we are from Chennai, we play cricket in the street, and in the beach. But obviously, as we come from India, Asian parents, so when you go to play cricket, I'll be always told off by my mother, you need to study first, because cricket is not going to pay you a salary. So study first, and then see how it goes. So I had to go to school, enjoyed my school, and I went to St. Mary's Matriculation Boys Higher Secondary School, which was around 1 hour journey from my house. So I had to take a bike ride to railway station, take a metro train, 30 minutes ride, and then walk again 15 minutes to the school. And by the time I come back from the school, I'm really gone. It's very tiring, that too in India with going on the metro train. But this is our school. It's a great school. I learned a lot. We had few friends, but unfortunately we don't have any connections anymore. A lot of friends went through different areas. So once I finished my school, I always had an ambition to get into medical school. And that's where my academic professional background started. The first journey is from Chennai to travel up to 500 kilometers to another city called Mysore to do my medicine. So the medicine, we did it in JSS Medical College. It's a cool college. Now it's become a automated university as itself. During that time, it was affiliated to Mysore University. It's a good college. And Mysore is a great city for, again, a lot of culture. It's a big palace, so we used to go always, a lot of our friends do, every Sunday night, the palace will be lit up. So we go there, spend a lot of time, have some street food, have a lot of food and enjoy. It was a good time. And it also has a lot of sculptures which are from more than 150, 200 years old. And of course, this is one famous restaurant I would like to share here. It's Hotel Triple R. So it's very famous for biryani. I hope you know what biryani means. It's a great food. And these are some of our close friend groups. And again, this was the only photo I was able to find from my circle. And this is how they looked after 10 years and after married. A lot of us were losing hair. I was still having hair at that time. We are still in communication with all of us. It's a great bunch. And most of them have gone on to practice in medicine. I'm the only one who deviated from medicine coming to this research field and teacher. And a big shout out to my close friend Jayant. He's a very famous ENT surgeon, orofacial surgeon back in India. He said he's going to join on Zoom. Hi, Jayant. So once I finished graduated my medicine, then obviously I wanted to start practicing. So the real life of a doctor, so until you're in medical school, it's all 9 to 5, finish your school, go back, have fun with your friends, study. But then once you get into the doctor life, that's where the real life comes from. I was so lucky to get a position as a senior house officer at Sri Ramachandra Medical College. And this was my mentor during that time, Professor Thanikachalam. So he looked pretty serious, and he's always serious. And he's so clever. And he's an interventional cardiologist, and he used to do 6 to 7 coronary angiograms a day. And then I used to follow him to the cath lab. And then during those days, there's no digital images, so they used to develop the image. And then we used to go to another place where we run this image and see what happens to the coronary arteries. And he will just say he'll just quickly look into all those images, and then come up with a diagnosis pretty soon. But on the first day when I went to the ward, with him. He showed me ECG, and then asked me what it is. I was not able to say. And then he said, go back, you're not coming to ward until you read this book. So I'm sure some of the cardiologists will know this book. So it's a specific book for ECG. So he gave me 1 week to go through most of the major topics in this book before coming to the ward, and I did. And that's where I started developing my interest in cardiology. And during that time, my brother was in Japan, and he had a connection with one of the professors there, who was doing cardiac surgery, who was a cardiothoracic surgeon. So I got an opportunity to do a PhD. So when I went to Japan, to be honest, I didn't thought I'm going to do research. I thought, oh, it's more like a clinical skill. Let's go and develop clinical skill, and then go back to India and continue my clinical journey. But however, that's where the life takes a twist. So in December 1999, I moved from Chennai to Japan. So Japan, it's a great country, but I was not in Tokyo. It would be nice if I had been in Tokyo, but I was far from Tokyo. It's again a 1 hour flight journey from Tokyo. It's called, it's an island consisting of 4 small cities. It's called Shikoku. Shikoku in Japanese is 4, so Shikoku Island. And I was living in one of the city called Kochi. So I started my PhD in Kochi, and again, Kochi is a city which has wonderful sceneries. It has a big port. It's a city of port. It's a great city, but it's a small town. I would say half the size of Dunedin, but the population were around 300,000 people. So it's still congested, but according to Japan, during that time they said, oh, it's a very small town, not many people around. It has a beautiful palace there, and Japan's fort are very famous. And the Kochi, this warrior is Ryomo Sakamoto. He was one of the famous warrior who was born in Kochi and was famous in Japan. He was a great warrior during the Edo period. And the trams, I love the trams in Japan. That's so good. I used to use trams to go everywhere. And of course, this food. This is a tataki. It's called tataki. It's a smoked tuna. So good. I love Japanese food, and this is one of the best food I have. But I turned into vegetarian now, so I don't eat any food. Pretty vegetarian. But I did enjoy a lot of food. Yes, that's where my actually research life started. I met Professor Shiro Sasaguri. He was my mentor who introduced me to research. He's a cardiovascular, cardiothoracic surgeon who actually, he was playing a big part in understanding or in establishing a minimal invasive cardiothoracic surgery. So when you talk about a cardiothoracic surgery, so usually what we do is the patients, when they have a problem with the coronary arteries, these are the arteries which is supplying blood to the heart. So sometimes they get blocked like this because of a diet or for any other reason, of a lifestyle or a disease. And the way we treat those patients is to have a bypass. So we try to get another blood vessel from either arms or the leg, and then connect it from the big artery to bypass this lesion and connect it to the downstream artery so that we can continue to supply blood to the heart. Because heart is pumping blood to every organ, but heart needs blood to keep its muscles stable. So he wanted to know, what happens is during surgery we stop the heart, and then do the surgery, and then we restart the heart. It's good, we are trying to reinstitute the blood, but at the same time what happens is when we restart the heart, it starts getting a lot of oxygen. Oxygen is good, but a lot of oxygen is not good. So it's going to induce a lot of reactive oxygen species that can damage the heart. So he was very keen on developing a therapy which can reduce this reactive oxygen species when we are restarting the heart. So we were starting to think about, okay, what we could do. So that was my first project with Professor Sasaguri. So we started thinking about how do we condition the heart. So can we actually condition the heart so that it can withstand big injury? What does condition means? So try to give the heart small injury, slow by slow, so that you get the heart to know this is what the injury is. And that's what we did. So on day 1, using some animal models, we gave small injury to the heart, and that activated some signals. And on day 2, when we gave a big heart attack to the heart, this injury was the heart, which was trained, which is conditioned, was able to withstand. So if you put this into the clinical scenario, when the patient comes to a GP with chest pain, often getting chest pain, they usually don't end up with a big heart attack. However, the patients who do not have any chest pain, most of the times they end up with a big heart attack. And so we looked into this. The main focus of our research during this time is to understand what is the mechanism which is actually protecting the heart from the injury by conditioning them. And we identified that one of the protein was, this is called BCL2. This protein reduces the cell death, so improves the heart function. And we found that this protein reduces the cell death, thereby protecting the heart from withstanding or making the heart to withstand a larger injury. And we were able to publish this. This was my first paper. I was so happy when I got this paper published. And of course, it didn't come straight away. This was after 6 rejections, I think. Obviously, we started from the bigger journal. But now this journal is pretty big. But during that time, it was a pretty small journal. But nevertheless, it was so good. We were so happy. And interestingly, my professor, Sasaguri Sensei is teacher in Japanese. He insisted that he doesn't want to be a lost author. The reason is, during that time, he says, sometimes, when you write the references in journal, they write first 3 names and then put et al. So if he goes as a lost author, his name wouldn't be there in the reference. So he always wanted to be in the second author. But he will be the corresponding author. And this was my first page on the thesis. So ideally, we had an option to submit the thesis report. In Japan, the thesis report is not like a big thesis like we write here. So it's more like you publish a paper, and then you write a report which is 4 to 5 pages. And this is the first page of my thesis report, which I wrote in Japanese. Obviously, went through a lot of corrections, but I managed to write in Japanese, and they said it was reasonably okay, I wrote. And I got through my degree. But the great thing is, during this time, and this is the group. This is the big department where, and hopefully you can find me. If not, that's here, it's me. And it was a great group. So we always take yearly photo, and that's exactly what we're trying to do in physiology today. This time, we're trying to take a group photo. So every year, we go for the end of the year dinner. We do some presentations, have some dinner, and we take the group photo. It's a great group. And I started learning Japanese here because during that time when I went there were only 6 Indians living in that city. Out of those 6 Indians, 4 were living with families, so 2 pairs. And it was only me in this university. The other one who was living alone was away from me. So the only way I can get along with people is start learning Japanese. And the way I learned Japanese is by watching TVs. So I'd go to the lab, come home, start watching a lot of serials, which I still continue watching Japanese serials. That's so great. I definitely recommend you to watch it. It's a lot of serials on Netflix. But the good thing is we publish a lot of papers, which is mainly focusing on how do we make the heart stronger after starting them following the surgery, so as a reperfusion injury. So only one paper I want to highlight here is this paper. We use one of the drug called MCI-186 to understand if it can actually protect the heart from this reperfusion-induced damage. The reason why I'm highlighting this paper is based on our results. They then did a clinical trial in Japan. So edaravone is another name for the drug, MCI-186. And they found they did this, they wanted to understand if this edaravone has some protective effect in the heart. And in fact, they found when the patients treated with edaravone, they had less enzyme, which is enzyme which damages the heart. And most importantly, they found these patients treated with the drug had a better heart function. The heart is pumping better. So for a researcher, this is a great achievement because when you see your research is being translated from bedside to the clinic, and we are so happy to see that it's actually, one, it was reproducible, but most importantly, it was reproducible in the clinic. So it was going well, and that's when the life took a turn. So again, coming from India, I was around 27, I guess, during that time. And my parents said, oh, no, you need to get married. No, I said, I'm still a student. No problems. You are 27, you need to get married. So that's when my story found its co-author. So that's Jayanti. I didn't see her during that time. The first photo I saw her, this is the photo. So I was sent this photo when I was in Japan, said, oh, I believe you like her. I said, yep, I like her. And I definitely liked her. And so I took a horse to go to the marriage. So this is our marriage ceremony where we come on horse. You can see the horse there. So got married to Jayanti. And since then, we have been having a wonderful life. Thanks, Jayanti. And we have been spending so much, lot of nice, good, great events, lot of great times, but also a lot of arguments. It's always good. It's always good. And you know who wins at last. And the great thing is, my professor, Sasaburi Sensei, he came to India to attend the marriage. And you could see his wife, she was wearing kimono, and she said it took her 4 hours to wear the kimono. You know, it takes time. So our marriage was starting at 6 o'clock. So she said she waked up at 2 o'clock in the morning to wear the kimono, and she came to us. And I was so, yeah, he liked me so much, and it shows that they actually came to India for marriage. And when I brought Jaya to Japan, she thought I was living in Japan, I was living a rich life, but she didn't realize I was living in a small home, which was actually a garage, converted into a single house. It is actually a garage, but we enjoyed. And she started learning, started enjoying the Japanese culture. And at some point, she also started working in Japan. So although she came from IT field, she started learning science, and she changed her field and went towards the science. So it was great. But at some point, we had to start, that's when I started graduating PhD. And then I met Professor Sato. He was in the same school. He was from the Department of Physiology. It's called as Department of Cardiovascular Control. And he saw me with Sasaburi Sensei once, because the work's still in Japan. So I go to work at 8 o'clock as a PhD student, and I stay there until 2 o'clock next day, early morning, come home, sleep, and then go back. So he met us at one day, which was around midnight, and said, oh, you're still working. And then we started speaking in Japanese. And the good thing is I picked up Japanese, and I was able to speak Japanese. So he said he actually offered me a lecturer position during that time. And that was my first academic job. And the first project we did together that's when the tissue engineering was picking up. He said, oh, I want you to create a tissue engineered heart. And we went to a university in Germany, in Hamburg, and we learned some technique, and we came back. In fact, I hope this works. And we actually managed to, we'll see. Never mind, you need to believe me. So that's what I tell my students always, sometimes. Not sure how it works. But we managed to create a tissue engineered heart, which actually was contracting. It was so good to see the heart actually contracting, and we enjoyed it. It was so good. And the other, sorry, I'm just going to go back, because I had another video I want to show you. So the other thing we did with Professor Sato is to develop a commercial system. Because if you remember the surgery, when we did the bypass surgery, we take an artery or a vessel and then do a bypass to make sure that the blood is flowing. However, one of the problems with this is we can't really image whether the flow is there, how the flow is, how the graft is working. So we came up with an idea of using a dye called indocyanine green. And this indocyanine green is a fluorescent marker. So when you inject this dye and when you use a particular wavelength in a camera, we can actually see this from the skin level. And then we have started collaboration with a company called Mizuho. So they developed this nice, beautiful camera and went into market and has been used in market. I think it's reasonably doing well in the market. So there was another system during that time, it's called HyperEye, but which can which can do the imaging only for a few seconds, whereas our system can continuously monitor it. And it's been often used like this in the heart surgery to understand whether the flow is good or the flow is not good. Then the surgeons can immediately repair it. And most often is used in patients who are undergoing breast cancer surgery, because one of the problems with the breast cancer is the metastatic lymph nodes. So the cancer can spread through the lymph nodes, but there was no way to understand whether it's spreading. So they're using this camera to understand whether it's spreading, and then they can remove the lymph node just by visualizing. So it's being widely used clinically. So it was a great thing. It was going well, but at some point we decided then, okay, this is the time we move on to the next part of our life, and that's where my son came in, Ashwin. So this is when he was around 6 months. So he went to India after 6 months, and this is how he came back. He loved Ampaman. It's one of the good characters. We had a good time in Japan, and he's now started playing cricket. He was best outgoing cricketer in the junior cricket in Northeast Valley Cricket Club. And now here he is. He's doing Bachelor of Oral Health, and he's going to US tomorrow for his holiday. Such a big boy now. So life was going good, but at some point we decided, yeah, okay, so the research was going good, but I wanted to do more research, more of a translational research. So that's when we decided, okay, we had to bid farewell to Japan. It was not an easy time, and thanks that all my colleagues came in to farewell us. So from there, our life took a next journey from Japan to UK, to Bristol. So to Professor Paolo Madedu's lab. So Paolo Madedu was a great mentor. He taught how do we need to do research, and he was always working so much into the Bristol Heart Institute. And that's when I started developing interest, understanding how diabetes affects the heart. So I want to understand what the link between diabetes and the heart. The reason is, as we know, diabetes is a global problem everywhere, including in Aotearoa, where we have 350,000 people living with diabetes, and another 250,000 who are undiagnosed. It is a big problem, and we know that diabetes is a major killer and the cardiovascular disease, the heart disease is the major killer in diabetic individuals. So the first we wanted to know during that time is, does diabetes cause any problem in the heart? So we went back to the preclinical model. We took a healthy and a diabetic animal, just did a cardiac scan over several months or several weeks to see what actually happens. So I have to show some data here. And compared to the blue line, what we see here, the pink line is the one which is a diabetic animal. I'm just focusing on these two. So these two data shows the cardiac function, how well the heart is beating or functioning. And as we could see here, compared to the healthy hearts, the diabetic hearts starting to show the difficulty. They're not pumping well. The heart function is going down as the disease progresses. But what we wanted to know is, when does this disease start at the molecular level? So we took some heart tissue samples at the early stages of diabetes when the heart was perfectly fine, heart scan did not show anything, took the heart tissue and then did some genetic analysis. And what we found here is, interestingly, even the echo at this point when the heart scan shows normal cardiac function, at the genetic level, at the cellular level, we found that the genes which are associated with survival, which is protecting the cells, were going down, whereas the genes which are associated with death is actually increasing. And that told us that definitely the heart disease in diabetes is not starting on the day you see the patient. So this is the day you see the patient in the hospital, but what we found is the disease actually starting much, much earlier in the diabetic heart. So we started looking at we did publish several papers looking at a lot of mechanisms, and just not the lab. So we had good time beyond the lab, I'm sure Pat would recognize this. It's a Clifton Bridge, so this is where we're living. And Clifton Bridge is very famous for a lot of activities, and that's a big famous balloon festival in Bristol. So we enjoyed a lot. It's also a good city. We had a good, and Ashwin was growing slowly. And at that time, I was going to a conference in America, and then on one night I received a call that my father has passed. He was diabetic, he was completely healthy. He just woke up one night and suddenly went into cardiac arrest. And that actually triggered me to understand, is there a way we can actually identify this disease before the problem starts? Because the patients with diabetes, they don't have any problem, they just end up with the problem with the heart attack or the heart failure. So that's when I started thinking about, okay, we need to find a biomarker of how we can actually diagnose this heart disease. That's when I saw this ad coming through. So looking for lecturer and senior lecturer in cardiovascular physiology. This was in 2011. So I thought, okay, let's apply, and wrote to Pat, and then I applied. And I got a call from Pat. Sorry, Pat, I modified this with ChatGPT, you're holding a phone. It was a good call. It was a good call. I still remember Pat was talking to me for almost like 20 minutes. And at the end of the call, Pat said, okay, I'm going to call you for interview. So you come over. Those are the good olden days. The department used to fund you to travel to Dunedin, spend here for a week, meet all the colleagues, and decide whether you are successful in the job, and if you are, you decide whether you want to take the job. So I came to Otago, and I told Jaya that, yeah, you know what, I got a call from New Zealand, I'm just going to go there, and she doesn't know that I applied for the job. And we never thought about we might be leaving UK. And I told her, I'll just go there because they're paying me. I'll go there, do my interview, and come back. But when I came to Otago, everything changed. It was such a beautiful, beautiful country. And Dunedin, when I met, and I know Regis, thanks Regis for coming today. Regis picked me up from the airport, and we have been traveling from airport to city, which was like, there's nothing in between, and Regis told a lot about the department. I met all the colleagues, and by the time I left, Pat unofficially offered me the job, and I was very keen to take it. Thank you, Pat, for offering the job. So we decided to move, and we made up our mind, so let's move. And that's where our life took a big turn to move from London to Otago. So when I came into Otago, my interest continued to understand, to find a new biomarker, to find some biomarker which can detect the disease before the patient develops a disease. So that's where I started developing interest on microRNAs. So these are small RNAs. And the microRNAs are getting famous. They have been, the discoverers of the microRNA received a Nobel Prize for physiology last year, and that tells how important the microRNA is in pathophysiology. And the way I try to make others to understand microRNA, if you've done biology, you know that DNA transcribes to mRNA, which then translates to protein. The microRNA act by either degrading the messenger RNA directly or inhibiting its translation. So microRNA controls how much protein you have. And protein is the main structural part of our cells. So I wanted to know what happens with the microRNA, again in diabetes. So Shruti, she joined me in the lab in 2013. And we started collaborating with Patrick. Thanks, Patrick, and Michael, and Sean. Sean was not able to be here. So the first thing she did is, and that's when Regis and Pete established Heart Otago, and we started collaborating with clinicians. So we started getting some samples from the patients who are undergoing coronary artery graft surgery, took some RNA, did some analysis, and what I can see here is there's blue and red dots. Red dots are from diabetic patients. So what we found is in diabetic individuals, all the good microRNAs are going down, bad microRNAs are coming up. Okay? So definitely show that diabetes is having some problem in the heart. And Shruti was more interested in understanding what happens to the vessels, blood vessels in the heart. And we know that in diabetic individuals, compared to the healthy individuals, the number of blood vessels goes down. And that's exactly what she found. So this is the diabetic group. She found that as the disease progressed, this is an animal model, the number of vessels goes down. But what was interesting is she also found that some of the microRNAs were also going down in diabetes. But more interesting is that these changes in the microRNA start much earlier before actually we see changes in the blood vessels, suggesting that microRNA could be a good biomarker to understand what's actually happening. The reason why they are good biomarkers is that they can be circulated in the blood circulation, so they can be released from the cells, remain in the blood circulation, so you can just do a blood sampling and understand what's happening. And that's what, again, Shruti did to collect blood samples from the diabetic individuals and then looked at the microRNA, and she found a lot of good microRNAs are going down as the diabetic individuals, as the diabetes progresses in the individuals. So she managed to publish some good papers, and right now she is in Harvard University going towards a tenure track position. And while blood is good, blood is still invasive, so we need to put a needle into the patient to collect the blood. So we wanted to see if there is any non-invasive samples we can do the microRNA analysis. So then we started looking at saliva. Saliva, we can collect it anywhere, and there was a study during that time showing that even saliva can express, microRNAs are also expressed in saliva. So we found, and we did a study with collaborator Keshava and Jaya, Lini, Bhagya, Daryl, and Patrick. So we found that some of the microRNAs are also expressed in saliva, and that's where my project is now going towards, to understand if salivary microRNAs can be a good biomarker for heart disease. It was going well, and obviously we thought, okay, Ashwin was lonely, so we thought, okay, he needs someone to play with. So we started thinking about another one, and now you can see the tummy is starting to become big, and that's where this girl, Anushka, came into our life, and she is the first girl in our family after 53 years after my sister, and you know how pampered she would be, and she is. And she plays cricket, she dances, and she said she wants me to put this picture because she likes it, it's more aesthetic. She keeps me busy all the time. So she came into our life, and the research was going, and we continue to publish. Thanks to the big team and all my students who came into my lab, so we managed to understand what's happening in the heart in terms of diabetes and how microRNAs are playing a role. Obviously, the next question is whether microRNA could be used as a therapy. And Nima and Andrew, Nima was a previous PhD student, so he identified that some of the microRNAs can be used as therapy. Answer is yes, microRNA can be used to protect the heart. We use this, and we also collaborated with Aniruddha in pathology, another student. So we also found that we can genetically modify the microRNA and protect the heart. And more interestingly, we also found that you don't actually need to inject microRNA, just get these animals to do the exercise. And you can see the third one is an obese animal, he's a diabetic one. So he started showing nice changes in the microRNA. So you don't even need to inject microRNA, just do exercise, it will change the microRNA and it will protect the heart. We managed to publish it in a very good journal. It was picked up by the editor during the review, referring to this journal, referring to this, our paper, saying that better start exercise much earlier if you want to make it easier, because if you leave it late, it's going to be too late. It was great, but then the microRNA seems to be good for therapy, but the problem is we can't just inject these molecules into the heart, because once you inject into the molecules into the heart, they just go away. And that's when Matt started his PhD with me. So we started looking at how, and Matt and Rishi, so we wanted to see, can we actually put the microRNA into something? And we started looking at the lipid nanoparticles, and these two, along with Alan and Shamal, so they developed this lipid nanoparticles. They put the microRNA into it, because once you put the microRNA into the lipid nanoparticles, they are stable for a long time, so that if you do one injection, they remain in the circulation for a long time, and they can make better protection. And this is now continued with Lijo, Jaken, and Shubhra, and they have been working on this project. But making this lipid nanoparticle is good, but still if you inject those lipid nanoparticles, they're still going to go everywhere into the heart. So what can we do? Can we put these lipid nanoparticles into a scaffold, make it like a Band-Aid, And that's where Jekin and Azam came into Azam's expertise came into effect. So we put these lipid nanoparticles into a bandage, so make it like a bandage for the heart, and that's what Jekin is working on. So, and he wanted to make a conductive bioscaffold for the heart, because he's trying to get the scaffold which can actually generate current, or which can conduct the current, and in fact he has done that in his preliminary studies, and he's trying to make this as a patch for the heart. So that's more about the research, but the other one which I love, thanks Daryl for introducing me to synchrotron radiation, is this big synchrotron radiation in Japan, where we go, have fun and work. It's a big radiation facility which allows us to image the small vessels in a live animal. So this is how the facility looks like, and what we can do is the small arteries. So these are the arteries which are supplying the blood to the heart, and we can go up to very tiny arteries, and we can actually measure the function of these arteries in a live setting. So the way synchrotron radiation works, we start work at 8 o'clock, work until midnight, and then go for food. So we eat, we continue to eat, we continue to drink. This is the normal beer, and this is the non-alcoholic beer. That's what I drink. This is how it looks. But it's a good fun. It's a great place to develop a lot of network, and thanks to James here, who was a mentor, who was a collaborator there in Japan. And although it's fun, it allowed us to publish a lot of papers. So we got a lot of papers out of this, and it's a great, a lot of papers, but also a lot of collaborations through those papers, just enhancing our network. Finally, this is my final data slide. And the other thing we are doing is, this is one of the argument I put forward for my RRR award. So one was what, like Julia said, it was about the saliva. So I changed my teaching lab from animal to using the student samples, saliva. It was well received. This is another one where Arib and Devin and others are helping me with this. So we are creating induced pluripotent stem cells, which, as we know, it can be created in any type of cells. So this is a cardiomyocyte. But most interestingly, we can make them into an organoid. So this is the cardiac organoid, which actually contracts. So the reason why we are doing this is we can just get the blood sample from someone, make these cardiac organoids, test a drug to see if it actually works. So that's where we are going now. And thanks to the university for recognizing this effort. So last week, it was a pleasure to receive the RRR award from Professor Cook. Thank you, Greg. Right, it was unexpected. So it was going very well when I had Pete in my office one day saying, "Oh, Rajesh, you should apply for the head of department." I thought it was a joke, and I didn't respond to him at all. But he came back after 2 weeks and said, "No, I'm serious, you should go." So I put forward my application. Thanks to Lisa for believing in me, for offering me this position. And I kept it simple as the head of department. It's a great department, so it's very easy. I try to be as positive and motivated, and try to keep up the morale of the department, transparent, and I trust my staff, and they trust me. That's important from my point of view as the head of department. Of course, we came up with a lot of challenges. We all went together. One of the biggest challenges we had was a management change last year, but great that we got it through. Tough external funding, but in spite of that, the department morale, I believe, it's still great. We're keeping it higher, but I wouldn't be able to do without these wonderful people who are helping me out. Rebecca and Regis started as a deputy HODs during my first term, followed by Rosie and Jeff. Thank you guys, thanks for always supporting me. And if you look at my professional background, I don't stay in the country more than 6 or 7 years. But in Otago, I have been here for the past 13 years. That's because of my family and fauna and colleagues and friends beyond work. So I'm a member of the Dunedin Indian Association. We do celebrate a lot of events, and I'm proud to be a member of this. I'm a member of the Hindu Swayam Sevak, sung, and I don't attend a lot, but whenever there's a big function, I try to be with the community. I also served as a president for Dunedin Tamil Society. So we did a lot of events as a Dunedin Tamil Society. So it's all about, it's not just work. This community which helped us to stay motivated and stay here in Dunedin. And a lot of our family come to Dunedin. This is Jaya's parents, my in-laws. So they came to Dunedin, and my family keep coming to Dunedin because Aotearoa is such a beautiful country. And of course, I try to grow my hair whenever I get my time. Thanks to Anushka helping with that. And of course, we play cricket again, because we are Indians. What else we do? Because of my passion to cricket, this is when I damaged my back, something with the back. That's when my sciatica started, so I don't play cricket anymore. However, I passed my level 3 umpire exams. So I do umpiring for the premier club games nowadays. I keep growing. I'm a Dunedin cricket umpire now. Right, so that's my last few slides. I need to thank a lot of people, starting from Shruti. Oh, I thought I was going to name each of them. Starting from Shruti, my first PhD student, Parul, Nima, Ash, Jason, Ramakant, Joshua, Eugene, Nilanjan, Prakash, Dhananjay, Rishi, Shalini, Christine, Matt, Lejo. Lejo just submitted his PhD thesis. We have still few more, Devin, Shwati, Jekan, and Arif going on, and one more coming through. I just accepted another course supervisor from Auckland University. So I think it's 22 now, still ongoing. I also like to thank my master's students, Bhagya, Emani, Scott, and Trishila. And also my honors students, Andrew, Hayden, Langi, Sophie, Ryan, Asha, Will, Anthony, Adeline, Lenny, Kualia, Eknur, Joseph, Michelle, Oakley, and Suhail. You made it. It's still going. And also my summer students, Betty, Jono, Langi, Sophie, Shreya, Amar, and Pearl, Mickey, and Has here. Is somewhere here, sitting here. And of course, my visiting students from US, they were here. And also my early career researchers, Deepthi, Amol, and Mickey, Sweta, Ingrid. Ingrid did a great job taking care of the lab. Shruti, Parul, and Jaya is now taking care of my lab, Dhananjay and Shubhra. Thanks to all of you guys. Without you guys, I wouldn't be able to do. But a big shout out to my admin team. Sorry guys, I didn't get your photos. I had to put a name. Shona, Anna, Chelsea, and Alice, and who is being taken care by Claire. Thank you, Claire, for taking care of them. And wonderful technical team was led by Sue, Caroline, Ash, Lorraine, Jean, and Joshua, an awesome healthy and safety officer, Rachel. Thanks, Rachel, for making sure we are doing everything appropriately. And clever finance advisory team. And of course, they say no money anytime I ask them. Sarah and Jody. And a brilliant purchase team led by Heather. Thanks a lot. And our outstanding teaching team, our science teaching fellows, and our med teaching fellows, they take care of everything. So I had an opportunity to do the teaching, coordinating, managing them this year, but they did everything by themselves. They can do everything. And of course, my collaborators, it keeps going. So all thanks to all my collaborators. So what I learned in science in the past few years is develop collaborations, develop networks. That's most important. So you develop network, and that will keep going. My international collaborators, my department collaborators, and my national collaborators. Thank you, everyone, because it's because of you I'm here today. And I understand that science is not just your idea. It's all about bringing it together and enjoying it together. Thank you so much. And then my last slide is thanks to our funders. Of course, without them, I wouldn't be able to do any of the research. And my last message, and our patients, because a lot of my research comes through the patients. And this is my take-home message for you guys. So we are doing a lot of research, but prevention. Okay? Prevention is much better than insurance. So thank you very much. Vanakkam, namaste. Thanks for coming along. Thank you so much.
Tēnā koe, Professor Katare, tēnā koutou katoa, ko Pete Jones tōku ingoa. Nō reira. Thank you all for coming along today. So my role is to thank Rajesh for his IPL today. Before I do, for those of you online who hopefully could hear the singing then and weren't sure what exactly that was, so that is a custom in New Zealand, so that's a waiata, and that's to tautoko, or to support Rajesh in a formal setting in respect of the lecture he's just delivered. So thank you again, Rajesh. So really this role is reserved for the head of department, and that's because it's quite a enjoyable role to do, to say thank you to a close colleague. It's probably one of the better roles of a head of department. But as Rajesh is the head of department, of course, it'd be a little bit disingenuous for him to thank himself for a great lecture. But irrespective of who's thanking him, I think anyone can stand here and also say thank you very much for tonight's presentation. It's always really fun to come to these and listen to what people have got to say, because you get to hear a bit of a different side about a researcher. You hear a lot about their research when they're standing there and they're passionate about what they work on, but often you don't understand how they got to where they are today. It's also really nice to hear from people whose background wasn't just science all the way through. It's great to see that the clinicians converting into researchers, although I do see there are some clinicians in the audience who still retain their clinical work, but also are very active in the research scene. It's also nice to see that journey through the different countries, predominantly around the Commonwealth. It's also good to see that in all of those cities, I think you've always lived by the sea. They're all port cities by the look of it, although you've got the very different climates of being on the beach, whether it's from the very, very hot south of India to, I guess, the hot south of Japan, but much colder Bristol and Dunedin. But throughout that time, whether it was that clinical focus or through the research, it's been clear he's had that focus on the cardiovascular disease and then moving into the diabetic effects on the heart. Hopefully what was clear as well is not only that passion for the topic, but also his pioneering use of the techniques. So a lot of that work in terms of the microRNAs or the stem cells is all very recent advancements. And I think it was a really fitting award to get that 3 Rs award, because really this is where science is going. And it's great to have a colleague in Dunedin, in New Zealand, doing the cutting edge of that types of work. Of course, it's not only being a researcher that's important at Otago to become a professor. That's looked at across research, teaching, and also service. I think some of those slides at the end just show the teaching aspect of Rajesh. You saw that number of students that have come through his lab. What's really nice is to see a lot of those students are actually in the audience at the moment, and that shows the quality of that mentorship and the fact they want to come and support Rajesh even though they've now left his group. From the service side of view, he's head of department. I think that says it all. This is often, I suspect, a fairly thankless role. He's getting people below him asking for stuff. He's getting people from above him telling him we probably can't have as much of that stuff. As he mentioned, he's done this through a really tough time, through a management of change, but he's done that with compassion and understanding, even losing some close colleagues. I now understand perhaps where some of those management skills came from, and also that hard work. Of course, you're having to put up with that when you're an umpire, so potentially on the cricket field you're getting more abuse than you do as head of department. But also that work ethic. Now, again, it puts into context how Rajesh has managed to run his research lab and be an effective HOD, as many of these HODs in the past have had to do as well. But that's getting those emails at 5 a.m, showing he's already getting up, doing what he needs to do, but perhaps he's not working as hard as you did in Japan. So it's been great working with Rajesh over that time. It's clear that Rajesh exhibits all of those things that we expect in a professor at Otago. So with that, all it does is leaves me to thank Rajesh again, and I hope you can join me doing that, and also to present him with a small gift to recognise the occasion. Tēnā koutou, tēnā koutou, tēnā tātou katoa.
And I get the final job of the evening. For those of you that don't know me, I'm Megan Gibbons and the Pro Vice Chancellor of Health Sciences, and I've decided we've got all the cool kids. So it's always really exciting to come to these and to hear the journey of our staff and where they've got to and that reflection back, because often when you're in it, you forget to have a think about where you came from and where you're going. So congratulations and thank you. And my final job tonight is to invite you all to the staff club to enjoy the evening with Rajesh and celebrate his inaugural professorial lecture. And I see him in the service role. I'm one of those ones going, no, you can't. But tonight, yes, you can.